All right, welcome back everyone to Unit 1, Chapter 2, and in this lecture we'll talk about basic chemistry. And so, once again, my name is Professor Travis Stroud, and you are in Anatomy and Physiology 1. So, let's just get started today. So we'll talk about some terms here. We'll define them very basically. And there's a whole list of terms in the learning objectives, and we'll go through all of them today. So the first one is matter. And matter can be defined as anything that has a mass and takes up space, okay? Pretty basic. All physical objects are going to be composed of what we call matter. An easily observed property of matter is its state or phase. And the classical states or phases of matter are, of course, solid, liquid, and gas. So if you're sitting at a desk and you knock on it, you can say that that's a solid form of matter. If you're drinking a bottle of water, liquid. If you've ever seen water evaporate, it turns into the gaseous phase, okay? So pretty simple there. And we'll talk about some basic chemistry, and we'll talk about more terms here. So the term element, we talked about that before. Element is the simplest form of matter that has a unique chemical property. So some examples are hydrogen, oxygen, and we have 92 naturally occurring elements on Earth, and 24 of them have roles in our normal physiology. Some of the really important ones: oxygen, carbon, hydrogen, nitrogen, calcium, phosphorus, okay, things like that. Atom, what's an atom? Well, we already talked about that in the first chapter as well. The smallest component of an element that retains the characteristics of that element. All right. This is going to be a little bit of a longer video, so I suggest taking it in pieces and, taking your time with it. Okay, let's talk about subatomic structure. So here is what an atom looks like. And if you remember, we said in an atom's nucleus in the center, we have things called neutrons and protons. Protons have a positive charge. You can remember that because it's pro. Like if you're pro something, you feel positive towards it. Neutrons has a negative, not a negative, a neutral for neutron, neutral charge, okay? And as we continue on here, in the clouds, electron clouds surrounding the nucleus, flying around, are electrons, and electrons have a negative charge. That's the subatomic structure of an atom. Okay, let's talk about using the periodic table. If you came up to the periodic table, you would see something that looks like this. And this first thing you would find is the chemical symbol. So this is the chemical symbol for carbon. If we were looking at oxygen, the chemical symbol would be O. Nitrogen, N. Okay, calcium, Ca. So some of them have one letter, some of them will have two letters. Okay, the next thing is atomic number. The atomic number. What is the atomic number? The atomic number is the number of protons. Then the next thing is atomic mass. Now, what's atomic mass? Well, atomic mass is actually the number of protons plus the number of neutrons. Protons plus neutrons. Okay? And so that's important to remember. Now, you might be questioning, why do we have 0.01 here? Well, I'll tell you about that in just a minute, but the atomic mass of carbon is actually 12. The atomic weight is a little bit more different than 12, but you'll figure that out in a second. So atomic mass is the number of protons plus the number of neutrons. Here are some examples of different atoms. Here we have a hydrogen atom, and this one, it has one proton, no neutrons, and one electron. Okay? 6 protons, 6 neutrons, like we said in carbon, giving the atomic mass of 6 plus 6 equal 12, and 6 electrons. Oxygen has 8 protons, 8 neutrons. 16 would be oxygen's atomic weight, and it has 8 electrons. Okay? All right, isotopes. Some basic chemistry of isotopes. Okay, what are isotopes? Isotopes are atoms of an element that have different numbers of neutrons And since they have different numbers of neutrons, they will therefore have a different atomic mass. Okay, so here's where we're talking about that 12.01 we just talked about. So if you see hydrogen right here, the one we just showed you, one with 1 proton, no neutrons, and 1 electron. But there's also deuterium, and that has 1 proton, 1 neutron, which is different, and 1 electron. And then tritium, which has 1 proton, 2 neutrons, and 1 electron. These are all isotopes of hydrogen. Now, they all act the same chemically. Okay, so they all act like regular old hydrogen, but they just have different mass numbers, okay, a different atomic mass. So do they have the same atomic number? Yes, they do, because they have all 1 proton. But they do not have the same atomic mass because obviously 1 plus 0, 1 plus 1, and 1 plus 2 are different answers. Here's what that looks like. Here's regular old hydrogen, here's deuterium, and here's tritium. All right? Okay, so then we talked about what a molecule was, and this is kind of review from the first chapter. Molecule is a particle made up of 2 or more atoms united by a chemical bond. Nitrogen being 2 nitrogen atoms. Glucose being 6 carbon, 12 hydrogen, and 6 oxygen atoms. And if the molecule consists of 2 of the same type of atom bonded together, it's called a diatomic molecule. So oxygen and nitrogen are diatomic molecules. If the molecule consists of 2 or more different elements, it is called a compound. So glucose, that's sugar, C6H12O6, is a compound. Carbon dioxide is a compound. These are diatomic molecules. These are compounds. Okay, ion. An ion is a charged particle, and a particle will get a charge when it gets unequal numbers of protons and electrons. Now, that makes sense because protons have a +1 charge and electrons have a -1 charge. If you've got +8 and -8, your overall charge is 0. 8 -8, 0, right? So, if you give up an electron or gain an electron, suddenly you either have a positive charge or a negative charge. If they have a positive charge, that means that they've given up an electron. In our example, oxygen has 8 protons and 8 electrons. If it had 8 protons and only 7 electrons, it would then have a +1 charge. 8 minus 7 is 1. So that would be called a cation. Now this is just something you have to remember. A cation is a positively charged ion. I like to say, well, if you are a cat owner, that's a positive thing. All right, hopefully that helps you. I like little things like that, little tricks. So cation is a positively charged ion. Anions are the opposite way. When you gain an electron, so if they had 8 protons and 9 electrons, suddenly it would have a -1 charge and be called an anion. And anions have gained an electron, they have a negative charge. All right. Electrolytes. So what are electrolytes? Electrolytes are substances that ionize, become charged in water. Things like acids, bases, and salts are electrolytes. They form solutions that are capable of conducting electricity. Electrolyte imbalance is one of the most important considerations in patient care. What things do electrolytes do? Well, they help you have muscle contractions. They help send nerve signals. And so electrolyte imbalance can either be make you feel very, very sick or even be potentially deadly. And so it's very important that you drink things like that have salt in them or that have other electrolytes in them, like Gatorade, for example, right? That's one that's always big. If you're being very active and you're sweating a lot, you want to replace those electrolytes. Some examples of electrolytes are sodium chloride, which is just table salt. When it goes into water, it becomes a positive sodium and a negative chloride ion. Calcium chloride becomes a 2+, so this is a cation that has a positive 2 charge, and there are 2 -1 charged chloride ions. Okay, can you see how a 2 times -1 plus a 2, just 1 plus 2 would equal out to be an overall negative charge? But yes, that is a calcium chloride turning into an ion. And then sodium bicarbonate turns into sodium positive and bicarbonate minus. So those are some examples of electrolytes. Okay, something called a free radical. What's a free radical? Well, a free radical is a chemical particle that has an odd number of electrons. When you have an odd number of electrons, that makes it unstable. Some examples are the superoxide anion, O2 minus and this little dot. And that dot indicates that there is an odd electron. When that odd electron wants to go somewhere, and so it makes this very unstable. These get produced, free radicals get produced by normal metabolic reactions all the time. Also radiation and also chemicals produce them. What happens is these free radicals will quickly combine with other molecules in the body, and that'll trigger chain reactions to produce more free radical production, and then that just starts destroying molecules. So free radicals, if they get inside your body and sort of start circulating around, that damage can be really bad. It can include cancer, heart tissue damage, and death and aging. And so free radicals are not a fun thing. You want to make sure that your body takes care of those. How does the body take care of those? Well, things called antioxidants, a chemical that neutralizes free radicals. Some examples that we produce are some enzymes, superoxide dismutase, or SOD. This is a enzyme because it ends in the letters ASE. That's something to keep in mind. And it gets rid of that superoxide, okay? It converts superoxide free radical into oxygen and H2O2, okay? A little bit different than H2O, obviously, but these are things that are not going to harm us, okay? And then things that you get in your diet, your dietary antioxidants, things like selenium, vitamin E, vitamin C, carotenoids. Carotenoids are pigments that you might see it right here, carrot. They're like orange and yellow pigments, carotenoids. So eating your vegetables, eating things that are high in vitamin C and vitamin E, very important. Remember, a molecule is a particle that's made up of two or more atoms held together by a chemical bond, right? So if you talked about O2, there's an oxygen, and they're held together by some kind of bond. That's called a chemical bond. So we're going to look at three types of chemical bonds in this lecture. The first is an ionic bond, the second is a covalent bond, and the third is something a little bit different called a hydrogen bond. And we'll get into that right now. So ionic bond, when you get an ionic bond, it is a bond between a cation and an anion. And you'll see them in things like salt, like sodium chloride. And these are very weak bonds, okay? They're not very strong. They easily break down in the presence of water. And you've seen that. Go ahead and take some table salt and sprinkle it into a cup of water, and all of that stuff dissolves. And that's because those ionic bonds are breaking up. You're turning it into positive sodium ions and negative chloride ions, okay? So these NaCl is held together by an ionic bond, okay? Just something that you got to commit to memory. A covalent bond is formed by when two atoms share electrons. So if you take a look here, this is a hydrogen atom with one proton and one electron, no neutron here. So it's not a heavy hydrogen. And the bond here, they are sharing electrons, okay? And what happens is that there are certain amounts of electrons that fill what are called valence shells. That's not something I'm going to ask you to learn for this course at this moment, but you should just know that these atoms are very happy to have two electrons in this shell. And in the shell, I mean in this cloud that are spinning around here. And so they'll fill this by sharing with each other, so that this sometimes has two of them and this sometimes have two of them, and it goes around so quickly that it's basically they all have two all the time. And this is a covalent bond, sharing electrons, okay? You can see this with carbon dioxide. Now this is a double share. You can see here, 1, 2, 3, 4. And these are called double bonds. So carbon dioxide has double covalent bonds. Okay, so pretty interesting stuff, but let's talk a little bit more about covalent bonds. There are two different types. The first is a non-polar covalent bond. where you get shared electrons that spend equal time orbiting around each nucleus. So equal time on this side, equal time on that side. And these are the strongest of all chemical bonds because they are very happy to continue sharing those electrons equally. And it's very good, very, very good, and they're very happy to do that. So these are very strong bonds, non-polar covalent. Now, polar covalent, they're polar because sometimes those electrons spend a little more time on one side than they do the other. And so the region where the electrons spend the most of the time, those electrons are negatively charged. And so if it's over there most of the time, that region gets the slight negative charge. The region where electrons spend the least amount of time, they have a slightly positive charge. And so if you think about a magnet, there's a positive end and a negative end. This is the same idea. Sometimes most of the time the electrons are on the left side, let's say. That becomes a negative side. Sometimes they're on the right side, but not nearly as much. That becomes the positive side. So the polar covalent bond, it is strong. It's stronger than an ionic bond, but it is not as strong as a non-polar covalent bond. Here's what that looks like. So this blue is basically at any point along the timeline, you could find the shared electrons in here. And you can see it is shared equally. Now, in an oxygen bond to hydrogen, this OH bond, oxygen is much better at calling the electrons over to its side. That is a term called electronegativity. Okay, electronegativity. And all I want you to take away from that is that oxygen is really good at pulling electrons towards itself. So you can see that most of the time, that electron that it's sharing with hydrogen is over on this side. So it has this little symbol, means partial negative, whereas the hydrogen gets a partial positive. Okay, now this polar covalent bond is really important when we talk about our next slide and polar molecules. So take a look from that last slide. Now there's two OH bonds. Now what is this molecule? 1H, 2H, 1O. This is water, right? Well, I give it away. It's right here, water molecule. Water molecules are made up of two OH bonds, which are both polar covalent bonds. Because of that, the entire molecule, let me go back. The entire molecule has a slightly negative and a slightly positive charge. Yeah, so it's a covalently bonded polar molecule because it has two polar covalent bonds. Gives them that slightly opposite charges on either end. One side negative, one side positive. That leads to an interaction called a hydrogen bond. Now, they're called hydrogen bonds, but I don't necessarily like, and I think a lot of people don't like the term bond because it's not an actual bond, it's an interaction. You see how these hydrogen bonds are these dotted lines, whereas a covalent bond is a strong black line? And that's because these molecules are not exactly bonded together. It's more that this partial negative charge is interacting with a partial positive charge. So think of it like when you bring magnets together that you bring the south pole and the north pole of a magnet together, they want to connect to each other. But if you don't let them connect to them, you kind of like hold them apart, you feel that pull, that connection. They want to come together. This is what a hydrogen bond is. It's a weak attraction between slightly positive hydrogen atoms, right here, and slightly negative oxygen or sometimes nitrogen. But we like to think of the oxygen because hydrogen bonds are really important in water. Water molecules form hydrogen bonds together because the partial negative oxygen connects to the partial positive of this water molecule, and this one happens over here. And this is really important for water. It's really important for life. It is why water sticks to itself, right? If you pour water out on a table, it forms a puddle and not just a bunch of water molecules all over the place that comes together because of these hydrogen bonds. It sticks these molecules together. It's why your blood can pump through your veins. It's really, really important for life. And so that's a hydrogen bond. Okay, let's keep moving on. We are now going to talk about acids and bases. So an acid is any compound that releases hydrogen ions or protons or H+ in the water. It's called a proton donor. So a hydrogen ion, a charged hydrogen, is also a proton. They are interchangeable words used for the same thing, an H+. So HCl will give up an H+ and a Cl-. That is a acid, hydrochloric acid. A base is a compound that will accept those protons, those hydrogen ions. Many bases will release OH- into the solution. OH- will then accept H+, and what is that going to build you? H2O, water. One example is sodium hydroxide, which is a base. Sodium releases this OH-, which then accepts hydrogen ions, okay? So let me just say this is probably a place where I would pause and come back later because we've talked about a lot, but let's continue on in this video and you come back when you're ready. Okay, a salt. A salt is an ionic compound that will not contain an H+, a proton, or a hydroxide, OH-, and it comes from an acid-base reaction. The basic salt that we're all pretty familiar with is table salt, sodium chloride, which dissociates into an Na+ and a Cl-. Okay, let's talk about an acid-base reaction. So, here is hydrochloric acid. Hydrochloric acid dissociates into a proton, the proton donor, and a chloride anion. Then we have potassium hydroxide. Potassium hydroxide, yes, K, by the way, is potassium. I know that's unfortunate. That dissociates into a positive potassium and a negative hydroxide. That's the acid, and that is the base, right? Proton donor and a donor of the hydroxide. Then we draw our arrow, and we see that H+ and OH- will turn into what? H2O, water. That is very good. The base has taken up these these hydrogen ions. Then the K+ and the Cl- will form a salt. A salt. Potassium chloride is a salt. So very nice. This is an acid-base reaction. The acid gives the proton, the base gives the hydroxide. That forms together to make a water, and a salt is formed. Okay, learning objective 2. If I gave you concentrations of acids and or bases, I would need you to identify their chemical composition, relative position on the pH scale, and do some examples. So let's talk about pH scale. You may have heard of it before, talking about, oh, this is acidic, or this is basic, okay, or this is neutral. So pH is the measurement of the protons, or the hydrogen ions, concentration in a solution. It is an expression, now stick with me because I know this is math and this gets everybody a little flustered, but pH is an expression of the acidity of a solution and it is derived from the molarity of protons. Now, pH itself is the negative log of the molarity of the protons. Okay. This is just something that we have to put to memory. pH is the negative log of the molarity of the concentration of protons. Now, what is really important here is that we figure out what the scale means. The scale goes from 0 to 14, with 7 being right in the middle. 7, a pH of 7 means neutral. It is a neutral thing that you could think of as fresh water coming out of a water bottle, is right around the pH of 7. Anything less than 7 is considered acidic, and anything greater than 7, 8 to 14, is considered basic, or also called alkaline. So acidic means acidic, obviously. Alkaline means basic. That's just another word you'll see. All right, let's take a look at this scale. Remember, pH is the measurement of the proton's concentration in a solution. Hydrochloric acid has a pH of 0. What that actually means is there is a lot of protons in it. It is extremely acidic. Hydrochloric acid will burn through your skin and burn through anything. It is extremely acidic, and it is only found pretty much in your stomach, along with other gastric juice, which is anywhere from 0.9 to 3. There are specialized mucus-covered cells in your stomach that allow this to happen. By the way, if you have ever had heartburn, that's why it burns so much, is because some of that gastric juice comes back through this valve and it burns those cells, and you feel how horrible it is because you don't want something that acidic. Lemon juice, interestingly enough, is very acidic. It is 2.3. Wine and vinegar, 2.4 to 3.5. Bananas and tomatoes, 4.7. You can see all this. Milk and saliva, just below neutral, so still slightly acidic, interestingly enough. Pure water, like I said, right about 7. And then we get into the basic side of things. An egg white is an 8. Bleach is a 9.5. Ammonia, 10.5 to 11. Oven cleaner, extremely basic, to 13.4. And sodium hydroxide, NaOH, a base, is, of course, extremely basic. Okay, so that's just something to put into your mind to start understanding what this looks like and what this means. So, some things that you might ask yourself. Use the pH scale from the previous slide and help you answer these questions. Between oven cleaner and vinegar, which is more acidic, which is more basic, which has a higher concentration of hydrogen ions, which has a lesser concentration of hydrogen ions, which has a higher pH, which is a lower pH. You got to make sure you understand the relationship between acidity, pH, and concentration of hydrogen ions. All right, let me know if you have any questions about that, and we'll move on. The third learning objective is we want to be able to associate the properties of water with their importance to the body. Water, of course, is the chemical formula H2O. It is a covalently bonded polar molecules because oxygen and hydrogen make polar covalent bonds. We also saw that hydrogen bonds form between different water molecules as a result of those polar properties. Why is that important? Well, I kind of gave you a little bit of a hint on that. It was because that gives water a set of properties important to life itself. The first is that water, it becomes a versatile solvent. What is a solvent? Well, it's the ability to dissolve other chemicals. You can see here, water is able to dissolve sodium chloride or salt. We've all seen that. Put some salt in the pot of water that you're about to boil and it dissolves. Hydration spheres, that's what this is called, a bunch of water molecules around a sodium and a chloride form, and it isolates the ions because of the partially negative charge. See, the red is the partially negative part, is pointed towards the positive ion, and the partially positive parts are pointed towards the partially negative parts. So that's what helps things dissolve. Water also its polarity leads to the things I talked about before. Adhesion, which is the tendency that one substance will cling to another substance. So adhesion, if you get water on your clothes, it doesn't roll off, right? Unless you've treated it with some kind of chemical, it obviously gets you wet. That is adhesion. And cohesion, when you pour water out of a glass, it all comes out together. That's because the molecules of the same substance, water, clings to itself. And adhesion and cohesion lead to two things that are very important: capillary action and surface tension. But I wanted to show you adhesion. Let's say this is the thread of your shirt. The water molecule sticks to it, and they also stick to themselves. Cohesion. I thought that was a funny little graphic. But surface tension is why you can get bugs walking on the surface of water. And it's because water molecules attract one another very strongly. And so as each molecule forms a bond with the ones in its vicinity, now what kind of bonds? Hydrogen bonds. That inward net force causes the molecules on the surface to contract and resist being stretched or broken. So you get things like this. Really cool. And then capillary action is important so that we can move water, blood, all things that are dissolved in water around. It's defined as the movement of water within the spaces of a porous material, like your veins and arteries and things of that nature, due to the forces of adhesion, cohesion, and surface tension. So you might get a question, is surface tension a result of adhesion, cohesion, or both? Well, this one, what is it due to? Well, it is due to cohesion. This is actually due to both. So, kind of interesting stuff there. Okay, now, water is something, has a a thing called thermal stability. And that thermal stability will help us stabilize our internal body temperature, help us maintain homeostasis. The first is that water has a very high specific heat. And the specific heat is the amount of heat that is needed. Heat is energy, okay? Don't think of it as a temperature outside. Oh, it's really heat, as you know, it's really hot today. We use those words interchangeably in colloquial language, but in science, heat is an energy that you need to put into something in order to raise the temperature of 1 gram of a substance by 1 degree Celsius. All you need to know is that water has a very high specific heat. Now, you know this because when you put a pot of water on the stove to make spaghetti or rice or something of that nature, it doesn't start boiling right away, right? It takes quite a long time, in fact, for it finally to start boiling. And that's because you have to put a lot of energy into water in order to raise its temperature. The next is that water has a high heat of vaporization. Heat of vaporization is exactly kind of what it sounds like. It's the amount of heat or energy absorbed by a substance that has to happen in order to convert it from liquid to vapor, vaporization. How much heat does it take to change water from a liquid to a gas? Well, it takes a lot. And also, when water does change from a liquid to a gas, it takes all that heat with it. And that actually is really important because when we get hot, what do? We sweat. And really that sweat is to cool us off because when that sweat evaporates, all that heat goes with it. Okay? So those are two things that help us, that water has, that helps us maintain homeostasis. Water is also something that is highly chemical reactive. It allows the ability to participate in chemical reactions. So water, like I said, dissolves and ionizes other substances. The example we gave was sodium chloride. And water itself can actually ionize from water, H2O, into H plus and OH minus, which is active in what we're going to learn about hydrolysis and dehydration synthesis reactions. Okay. Whew, might be a good place to take another break and then come back now for learning objective 4. Where we will identify subunits, examples, and general functions of organic compounds and processes by which they are metabolized. All right, organic chemistry. Now, trust me, we are doing the absolute bare minimum of organic chemistry because that class is quite difficult in my experience as well, so don't worry, but we'll make it through this. Organic chemistry is defined as the study of organic compounds containing carbon in what are called covalent bonds. There are 4 types of organic molecules that you will put to memory. Carbohydrates, lipids, proteins, and nucleic acids. All part of a healthy, balanced diet, right? First, what is the difference between a monomer and a polymer? Well, this example here is a monomer example. So monomers are essentially the building blocks of larger molecules. The smaller subunits that when you put them together, you form a poly or many, mer meaning unit. 1 unit, many units. A polymer is a large macromolecule which you get by binding monomers together. So you take these Legos and you build this big Lego castle. You take monomers and you build polymers. All right? So the first we'll talk about is carbohydrates. And the basic formula for carbohydrates is CH2O and N, where N would be the number of carbons. So if we put 6 here, it would be C6H12O6, right? That's glucose. But we could also put a number of other numbers here, which would be different carbohydrates. These carbohydrates are variably sized. You might have larger molecules called polysaccharides, like a starch or glycogen or cellulose. All of those are polysaccharides. You know what starch is. Glycogen is within your cells, and cellulose is within plant cells. And you can have very simple carbohydrates, monosaccharides, like glucose, fructose, and galactose. Glucose is right here. Fructose is a little different. Galactose is a little different. But those are monosaccharides. You put many glucoses together to form polysaccharides. Here's what that looks like. There are glucose monomers and, let's see, no. Here are, this should say probably polymers. many monomers put together. Here are the monosaccharides, glucose, lactose, and fructose. You can see they have this characteristic pentagon shape, and if you put many of them together, you can have a nice polymer, a large polysaccharide. Okay, now we'll talk about lipids. Lipids, aka fats, aka things that they told you at once you shouldn't eat, and now they say you should eat, and okay, we'll talk about this. Lipids, there are 5 primary types of lipids in humans. Fatty acids, triglycerides, phospholipids, eicosanoids, and steroids. Now, the general functions of lipids are energy storage. Remember, if you, like me, need to hit the gym just a little more than you actually do, we have a lot of energy stored in fat. Thermal insulation, for those of you that aren't as lucky, I'll say, to have some energy storage like I do. You get a little colder than I do in the winter. So thermal insulation, cushioning. I don't hurt my butt when I sit down as often. But yeah, cushioning is very important. Cell membrane structure, steroids here and phospholipids, they are very important in cell membrane structure. And then chemical signaling between cells. All of these things are lipids, are uses for lipids. And so lipids are very, very important. Let's talk about what they look like. So, so they are long chains of carbons, okay, and hydrogens. You sometimes hear them called hydrocarbon chains, okay? And there's one, and here is a, what do you call these? You call these phospholipids. They've got a hydrophilic region, meaning hydrophilic means water-loving, so water can go on this side, water will stay away from this side. You put all these together, you make cell membranes, okay? And here's a steroid, and how do I know that? Well, it looks like an S. See that? If I drew an S here, that would be a steroid. So just stuff to look at. Okay, but a triglyceride is one that I'd like you to remember. It is a lipid, and its monomers are fatty acids and a glycerol. In fact, 3 fatty acids. You can see them right here. Here they are. 1, 2, 3. And a glycerol, this in blue right here. So 1 glycerol, 3 fatty acids makes a triglyceride. Okay, that's a lipid. Now we'll talk about proteins. The protein monomers, the monomers of proteins, are amino acids. There are only 20 amino acids that are used to make all of the different proteins. It's really quite interesting. If you ever got into talking about how your cells end up making proteins, it was very interesting to see how the body puts together all these different, only 20 different amino acids to make everything about life. Very cool. So let's talk about amino acid structure. So an amino acid has a amino group, which is NH2, N, 1, 2, 2 H's. It then has a central carbon group that is also attached to a carboxyl group. C double bonded to O, single bonded to OH. That's a carboxyl group. That's an amino group. The last group is called the radical variable group, or the R group, as I like to call it. So the R group, look, if you got rid of all the pink on every one of these 4 slides, you would see an amino group, a central carbon, and a carboxyl. Amino, central, carboxyl. So though that does not change, what changes and what makes amino acids different from all the rest are the R groups. If the R group changes, it changes what the amino acid is. Okay, so that's what changes amino acids. Now, you put a bunch of amino acids together in a chain, and you make a protein. Very simple. Okay. Functions and examples of proteins. So structure could be a function. Things like keratin in your hair, your skin, and your nails. Collagen in your cartilage, which keeps it stretchy and in the deep layers of your skin. Communication, things like hormones and hormone receptors are proteins. Membrane transport proteins, things that push things in and out of cells. Metabolism, things like enzymes are proteins. Yes, enzymes are proteins. And protection, antibodies, clotting proteins. Proteins are very, very important. Let's talk about the levels of protein structure. So the first is the primary structure. And the primary structure of protein are just a sequence of amino acids. So you could have, let's just call them by the color: blue, red, yellow, purple, purple. That's the start of this amino acid. That says polypeptide. We call it a polypeptide because the bonds that stick two amino acids together are called peptide bonds. So you put a bunch, you get a polypeptide. But this is a protein, essentially, okay? And one protein could be blue, red, yellow starting off. Another could be purple, yellow, green, okay? It's all based on the sequence of the amino acids. The secondary structure refers to local folded structures that form within the polypeptide chain due to the interactions between the R groups, okay? Now, the most common things here are either the alpha helix, so either they start spinning like this, or they fold into a beta-pleated sheet. So alpha helix, beta-pleated sheet is secondary structure. Tertiary structure is the overall three-dimensional structure of the polypeptide, and I said that this was due to R groups. No, this is just due to interactions between the molecules. The R groups, if we went all the way back here, interactions between the R groups is what makes the overall three-dimensional structure. Okay, so you can see how here is an alpha helix, here's a beta-pleated sheet, here's another alpha helix, right? Things of that nature. You put all of that together, you look at the entire thing, and that is the tertiary structure, the overall three-dimensional structure of the polypeptide. Okay, sometimes you stop there, and that's your entire protein. But sometimes proteins are conglomerates of two or more polypeptides coming together. One example I could think of is what carries oxygen around to your heart. Okay, not to your heart. It carries oxygen in your red blood cells. Hemoglobin. Hemoglobin is a protein that is a conglomeration of multiple polypeptides. And if that is true, if there's an association of two or more polypeptide chains, we call that quaternary structure. All right? Nucleic acids. What are the monomers of nucleic acids? They are nucleotides. And so sometimes nucleotides and nucleic acids, those things run together. But they are the monomers of nucleic acids are nucleotides. Nucleotides are made up of three things: a nitrogenous base, a 5-carbon sugar, and a phosphate group. Now this is ATP. ATP is adenosine triphosphate. Adenosine triphosphate. Triphosphate. 1, 2, 3 phosphate groups on ATP. But other examples of nucleic acids are DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Functions of DNA and RNA, obviously, are your genes, heredity, and things like protein synthesis. So you go from RNA to protein, okay? You start with DNA, make an RNA, and then you make the protein. Okay, very cool stuff. Here's just an example of DNA. And if we look down, you can see here are the nucleotides, the phosphate group, the 5-carbon sugar, and then the nitrogenous base. And in DNA, there are A's, G's, T's, and C's. A's and T's go together, G's and C's go together. And that's the only way in which it happens. A's don't go with C's, and G's don't go with T's, or vice versa. All right? If you ever wanted to remember that, and it's not something you need to learn now, but at Garden City, just how I remember it, helps me out quite a bit. Let's talk about ATP, adenosine triphosphate. It's a very important, kind of an understatement there, and well-known nucleotide. That specific nucleotide is not a monomer of DNA or RNA, but it is in fact the energy dollar of the cell. And essentially, if you need to do work in your cells, you need to use ATP. Now, you can see how this little squiggly line here connects one phosphate group to the next. This is to denote a high energy bond. If this bond breaks, a lot of energy comes off of this, when the cell can then use that energy to do work. And so ATP is used to do cell work. So you can think of it as the energy dollar of the cell. Okay, how do we turn polymers into monomers? So how do we cut them up, essentially, right? That's how you have to do it. You start with a starch molecule, and you end up with a bunch of glucose molecules. We do that by hydrolysis or hydrolysis. Lysis means to cut. Hydro is water. And so that's exactly what it is. It's a breaking up of larger molecules into monomers using water. It's also called a decomposition reaction. The example, you have AB, you add water, you get A plus B. Right here. Here's our dimer, just means 2 units, 1 and 2. Combined here of the bond, you add water, it splits to H plus and OH minus, and you get an OH here and OH here and 2 monomers. Decomposition reaction or hydrolysis. How do we join monomers? How do we go the other way? We call that a dehydration synthesis. Take away water and create something. That's what it means. And that's exactly what it is. You've got 2 monomers. You then you take off this H and this OH, you produce water, and you combine your 2. So you take your amino acids and go to this protein molecule. That is a dehydration synthesis. It is also called a condensation. Condensation because you're making water, or you're condensing these 2 things, whatever way you want to think about it. And it is the exact opposite of hydrolysis, right? You go from A and B, you take away water, and you are left with AB. So pretty simple stuff here, but just stuff you need to put to memory. Okay, learning objective 5. Explain how enzymes work and why they are important to the body. So proteins also function as biological catalysts. And when they do that, they are known as enzymes. What's a catalyst? A catalyst is something that causes or allows a chemical reaction to occur quicker than it would by itself. So how do they do this? They do it by lowering what is called the activation energy. You can see here. Here's before we add an enzyme to some kind of chemical reaction, and here's after. So in order to get a chemical reaction to go, the body has to put in some amount of work to get everything ready, and then it will go. That's a lot. But if we put what the enzyme does is it grabs things, puts them in just the right order, in just the right connection length, in the right space, direction, and so it takes far less energy to get something to happen. Okay? So how do enzymes work? Well, enzymes are substrate specific, and what that means is that a specific enzyme only works on a specific substrate. You can think of it as a lock and a key. This is the key. The substrate is your key, the enzyme is your lock. One key only goes into one lock. So sucrase enzyme is the enzyme that will take sucrose, which is just table sugar, and break it down into glucose and fructose. So what happens? Here's the active site. The active site is just the very specifically shaped part of the enzyme that binds its substrate. Sucrose is the substrate. Sucrase, ASE again for enzyme, binds. This becomes the enzyme substrate complex. The chemical reaction occurs, and the enzyme and the reaction products are produced. Are the enzymes consumed by the reaction they catalyze? No. The enzyme remains unchanged, and it can go back to the top and get ready to do this again. All right? So, things that you need to know. Can you identify the enzyme in this example? Well, I'll give you a hint. It's the blue one right here. That's the enzyme. This will then be the substrate. Where's the active site? Right here. The active site very specifically matches right here, this substrate. The enzyme substrate complex when they come together. And the products? Well, the products are here. We've now taken A, B, and made A and B, for example. Here is our enzyme unchanged at the end. All right. So how do changes in pH and temperature affect enzyme? Well, every enzyme will exist in an ideal place with both pH and temperature. And any change outside of that ideal range will change the shape of an enzyme. We call this denaturation. Now, remember we said in the very beginning, form is tied to function. Form is tied to function. Well, if you change the form, you're going to change the function. So the change in the shape of the lock or the active site so that the key, the substrate, won't fit. So if that happens, you're not going to get the result that you want. The reaction will not occur. All human enzymes are designed to function optimally, produce the fastest results near body temperature, 98.6 degrees. This is why fevers or getting very, very cold is a bad situation, because your enzymes will start to change shape. Now, different enzymes also will have different optimal pH levels depending on where they live, essentially. So here's our enzyme right around 37 degrees Celsius. You can see its activity is the highest. And if we start getting too high of a temperature, you can see that falls off really fast. And about 113, well, this is Celsius, but that is a cutoff of where you are not going to be living anymore. You can get colder, but not too cold, or else you also suffer the same fate. Now, pH is a little bit different. Pepsin occurs in your stomach, so its optimal pH is very low, around 3. Right? That makes sense. Salivary amylase is in your mouth, so right around 7 to 6.6 is where its functional optimal pH is. And then trypsin is in the small intestine. And the small intestine is a little bit more basic. And so its optimal pH is right around 9. Okay, so there's different optimal pHs, certainly, but the temperature is body temperature, the ideal. Okay, now is where I would stop, and you'll come back to me in the next video for chapter 24.